And my co-author is Laurent Metzinger from Paris. So first, let's talk about RNA-based therapies. As you certainly know, it is a therapeutic field in full growth. Can you see my mouse?
Yes, sir.
Okay. So here I have shared the link from INSERM in France. INSERM is the French National Institute for Health Research. And there's a whole file on RNA-based therapies. So the first RNA-based drug called fomivirsen was in fact introduced in 1999, right, in the States and Europe. This was against cytomegalovirus retinitis. And the first small interfering drug was commercialized in 2018 by FDA. It is called patisiran, and it's against transthyretin mRNA. As you have all certainly heard, there are actually two RNA-based vaccines that have maybe made available against COVID-19, so Moderna and Pfizer and several others against the epidemic of COVID-19. Several other vaccines, mRNA vaccines, are under development against flu, Zika virus, and so on and so on. So the message here is that many RNA-based therapies are in development in the world for different diseases and with several techniques that I will explain later in my talk. First, some words about RNA. Most of the people have heard about messenger RNA, but there's a lot of other RNAs. The take-home message here, important thing, is that there are several non-coding RNA. So non-coding RNA, as the name indicates, are non-coding, so they do not code for proteins, but they are functional. So they are subdivided into two classes: small non-coding RNA, also called microRNA, or long non-coding RNA, LNC RNA. The small non-coding RNA are small, approximately 20 to 25 nucleotides. They are transcribed by RNA polymerase of type 2, endogenously processed by endonucleases, and well conserved about tissues and through species. They were discovered in 2000, and we now know that in humans there are approximately 2,000 microRNA. More recently, in the last years, the high throughput sequencing techniques have shown us that, contrary to what is believed, most of the genome is transcribed. When you look at textbooks, they say that approximately 3 to 5% of the genome are transcribed, mainly to messenger RNA, transfer RNA, ribosomal RNA, and so on. We now know that it's actually false, since more than 80% of the genome is transcribed. This will give a lot of new RNA species that are not really well characterized, called long non-coding RNA. Approximately now 30,000 RNA are known. They are longer than 200 nucleotides. They are also transcribed by RNA polymerase of type 2, mostly 5' capped, polyadenylated, at least in parts on plate. What is interesting is that they are poorly conserved, so they are more specific for tissue. Here are some of the species known. So when you look at RNA, you have the coding RNA, so the 25,000 messenger RNA. You have also small nucleonar RNA. You have interfering RNAs that are short, including microRNA, which are the most well characterized and will be the subject of my talk. You also have circular RNA, and this 30,000 long non-coding RNA subdivided into several branches, and also, let's not forget, transfer RNA and ribosomal RNA. I will now talk about microRNA. So these are the short non-coding RNA. They are non-coding single-stranded RNA. They regulate post-transcriptionally almost 2/3 of the genome, and they are implicated in numerous physiological disorders, such as oncogenesis, metabolic syndrome, inflammation, and so on. They are carried in the blood via microvesicles, exosomes, endothelial apoptotic bodies, AGL, and so on and so on. Concerning biogenesis, microRNA are in fact synthesized in our nucleus. So they are endogenous. So you have first a precursor that is called primary RNA, that is transcribed by RNA polymerase of type 2. It is cut by an RNase called DROCHA with coactivator GGCR8 into a smaller species called primary RNA. We are still in the nucleus. Primary RNA will be exported in the cytoplasm via this protein called exportin 5. After that, it will be taken into charge by this other complex called DICE-RISC, RNA-induced silencing complex, which has an RNase called DICER, other proteins called argonauts, and so on. RISC will take this microRNA to its messenger target mRNA, and this will, in fact, be actually an imperfect sequence homologic pairing, which in fact means that the target mRNA will be, in fact, less transcribed. So microRNA in general are repressing expression of the proteins in the ribosome. Why are they so interesting? It's because they are innovative circulating biomarkers for diagnostic of prognostic of several diseases. Concerning kidney diseases, we were among the first in the world to show that microRNA are biomarkers for chronic kidney disease. Also, they are implicated in other kidney diseases, such as diabetic nephropathy, PKD, kidney fibrosis, IgA nephropathy, and so on. So I will now talk a bit about chronic kidney disease. What happens in kidney disease is that the kidney is less functional because the nephron function less, and that means that uremic toxins such as inorganic phosphate, indoxyl sulfate, etc., accumulate in the blood. When they accumulate in the blood, you get oxidative stress, inflammation, that will give bone troubles, but also cardiovascular diseases such as arterial hypertension, atherosclerosis and arteriosclerosis, ventricular hypertrophy, and vascular calcification. This will in fact lead the patients to end-stage renal diseases and an increased cardiovascular mortality. So since 2009, we have studied microRNA's role in CKD, in vascular calcification, atherosclerosis. We have shown that they can be innovative biomarkers, but also innovative therapeutic targets. When you look at a vessel, you have here the arterial wall. So in a big vessel, that is subdivided into endothelial cells, so the intima, vascular smooth vessel cells, the media, and the adventitious pericytes. In the CKD process, uremic toxins accumulate. You have the deposit of calcium phosphate, inflammation, oxidized LDL will stick to the vessel. So you have endothelial dysfunction in the intima, and in the media, the vascular smooth muscle cells will be changed. They will be transdifferentiated into osteoblast-like cells. It's like if the patients make bone in the vessels. And that is, this phenomenon is called vascular calcification. We studied the role of microRNA in this process. We developed several projects. On the left, you have an in vivo project in mouse models of CKD and also atherosclerosis. On the right, you have in vitro cell lines. In the mouse, we studied the transformation of VNC, of vascular smooth muscle cells into osteoblast-like cells in the aorta. Also endothelial dysfunction, leading to vascular calcification. We looked at microRNA in the tissue, but also in the serum. Considering smooth vascular cell lines, we looked at vascular smooth muscle cells transdifferentiation. So there's transformation into the osteoblast-like phenotype. We looked at osteoclastogenesis because this is the rebirth of osteoclastogenesis. And also we looked at endothelial dysfunction, looking at endothelial cell lines. We looked at several microRNA, the main ones being microRNA 126 because it is endothelial cell specific and implicated in angiogenesis and endothelial dysfunction, but also microRNA 223 because it is implicated in inflammation, but also osteoclast differentiation and vascular smooth muscle cell transdifferentiation into the osteoblast-like phenotype. I will talk today about the in vivo project. So the mouse model is here. It is a CKD model where you take mice, you electrocoagulate the right kidney at 8 weeks of age. So one kidney is non-functional. You let them recover for 2 weeks. And after that, you make an nephrectomy of the left kidney. After this process, only 1/6 of the renal function remains. It's called the 5/6 nephrectomy model. These mice develop CKD quite rapidly. So that was our first model, CKD mice, compared to the sham control. On the other hand, we also took ApoE knockout mouse that developed atherosclerosis. This ApoE knockout mouse were also subjected to the CKD process. And ApoE knockout mouse with CKD developed vascular calcification. In these mice, we took blood samples and we sacrificed them after 2, 4, and 10 weeks, and we took the aortas of the tissue. When you look at the aorta, so the tissue, we found that when you look at microRNA 2 and 123, so an inflammatory microRNA, you have an increase of the expression of the microRNA in the tissue that is increasing with the number of weeks in CKD. After 10 weeks of CKD, you got a doubling of this microRNA in the wild-type mice, but also a tripling in mice that have CKD and ApoE knockout, so they are both atherosclerotic and in CKD, they develop vascular calcification. So that was for the microRNA. We looked at messenger RNA that are targets of this microRNA, and I will remind you that as a microRNA increases, its target is expected to decrease. This is indeed what we found by Western blotting. So that is 2 targets of microRNA 223, the transcription factor NF1A and the glucose transporter GLUT4. For both of these targets of microRNA 223, we found that it was in fact the both proteins were decreased as the microRNA increased. So that confirmed our hypothesis that microRNA 223 is implicated in the CKD, atherosclerotic, and vascular calcification processes. We next looked at expression of this microRNA in the serum, in addition to what we saw in the tissue. So here we found the reverse, meaning that as microRNA increases in the aorta, it decreases in the blood. That was true for both microRNA 126, so the endothelial specific, and for microRNA 223, so the inflammatory ones. At all days of uremia, we found a decrease of the expression of microRNA 223, microRNA 136 in the blood. And I will explain in the later part of the talk that we confirm these results in the big cohort of human CKD patients. So to summarize these studies, we found that the arterial wall, that microRNA will, in fact, increase in vascular smooth muscle cells, transforming these vascular smooth muscle cells into the osteoblast-like phenotype that will make, in fact, the aorta to develop a bone-like structure with this calcium and phosphate, inorganic phosphate deposit. And on the other hand, we also found that microRNA will decrease in osteoclast-like cells. So these osteoclast-like cells are in the vessels to fight to counteract the ossification. MicroRNA 223 will decrease the number of these osteoclast-like cells. So both processes, so increase of osteoblast-like cells and decrease of osteoclast-like cells, will in fact trigger vascular calcification in CKD patients. So we have hypothesized that modulation of microRNA 223 expression could potentially be a new therapeutic pathway to cure vascular calcifications. For this, in collaboration with a team in Paris, so in term U966, Dr. Anne-Marie Lompré at Pitié-Salpêtrière, we looked at the modulation in vivo of microRNA 223 in a rat model of cardiovascular disease, namely angioplasty due to the balloon injury in the carotid. This is a model where you insert this balloon in the carotid, make an angioplasty. We first showed that the expression of microRNA 223 is markedly increased in rat carotid during angioplasty. We next developed adenoviruses, recombinant adenoviruses that were in fact designed to either increase microRNA 2 expression called pre-microRNA 223, or adenoviruses that are in fact developed to inhibit expression by sponging the microRNA. So pre-microRNA increase, sponging decrease, inhibition. Here is what we found in the carotid of the rat. So this is a control carotid without any injury. In the angioplasty model, you have in fact this invasion called restenosis of the carotid, so one carotid. When we increase microRNA 223 expression, we found that there was no effect, almost no effect on restenosis. But on the opposite, when we sponge microRNA by putting an adenovirus able to inhibit microRNA 223, we found a marked decrease of neointima, meaning that inhibiting microRNA 223 could be a new way to, in fact, protect the aorta for neointimal hyperplasia. To summarize this finding, in a restenosis model after carotid artery on balloon angioplasty, after 14 days, you have a restenosis. When you increase the expression of microRNA 223, you have almost no effect. But when you decrease microRNA 223 expression by sponging its expression, you have reduced neointima in hyperplasia. So that is a clue to have a new therapeutic model. In parallel to this study, we looked at a more fundamental question. How does microRNA 223 regulate gene expression in a cellular model? For this, we used a cell line called Rho cells 264.7, which is in fact a monocytic cell line. This monocytic cell line can differentiate into multinucleated osteoclasts, but also into type 1 or type 2 monocytes, depending on the cytokines you feed it with. We looked at these cells. We induced either an increase of microRNA expression using pre-microRNA 223 constructs, or we inhibited its expression using anti-MIR 223 constructs. That is for 48 hours. We next made several samples that were used for transcriptomics using Affymetrix microarray, transcripts that were used for proteomics using nano-LC MSMS, and transcripts used for metabolomics using NMR. The aim was to identify as much as possible the complex gene regulation responses to various stimuli, and to determine as exhaustively as possible the way a given microRNA, so here microRNA 223, exerts gene regulation function in the Rho cells. Here are just some of the results we found. So concerning transcriptomics, we found that microRNA 223 changes approximately 120 RNA expression, including 30 long non-coding RNA. Here you have the proteomics results in this heat map, and we found that 52 proteins were significantly deregulated in proteomics. Using gene ontology for both transcriptomics and proteomics results, we found that the proteins on mRNA change were associated with cell death, splicing instability on messenger RNA, bone remodeling, and metabolism, with a central role for the inflammatory cytokine NF-kappa B. Using Western blotting, we confirmed some of the proteomics results and found significant changes in, for example, CARM1, UB2G2, cactin, and NUFA-F6 during macrophage differentiation, but also osteoclastogenesis. So these proteins could be potential biomarker of these processes. Considering metabolomics, so that was studied using NMR, we found that several metabolites were in fact changed, and these metabolites will alter the metabolic profile of cells and may affect again their apoptotic and proliferative cells. Here's a heat map where we have all the significant metabolites. To summarize this finding, we found that when you change the expression of microRNA 223 in the monocyte cell line Rho 7, you have changes in transcriptome with, for example, several mRNA that will be affected in proteome, so for example, cactin, CARM1, etc., and in metabolome. These changes will be reflected when you differentiate the cells into either macrophage type 1, type 2, and osteoclasts. A central role of NF-kappa B was shown with effects on programmed cell death, RNA regulation, metabolism, and so on. So that gave us a base of knowledge to better understand the way this microRNA affect gene regulation in a cell type that is able to differentiate against to macrophages or osteoclasts. So we will develop these as future microbiomarkers for CKD and so on, example cactin, CARM1, and innovative therapeutic targets for clinical use. Also, this enabled us to identify key components in macrophage differentiation activation, so define novel targets to modulate inflammation, host defense, vascular calcification, CKD. Finally, a study of these two microRNAs, so 223 and 126, as biomarkers of the CKD process. So this study was made to evaluate the predictive value of the levels of two circulating microRNAs, so in the blood, 223 and 126, that was in a really big cohort of 600 CKD patients at various stages and 27 healthy volunteers. The aim was to evaluate the association with cardiovascular renal events or mortality over a 6-year follow-up period. This was done in collaboration with UTOX network and the University Hospital of Ghent in Belgium, nephrology department. So CKD, I didn't say it yet, is divided into 5 stages. So from stage 1, which is a milder one, to stage 5, which is a most invalidating one. And finally, you have stage 5 CD, where patients are in hemodialysis. So we had patients at all stages of the disease, and we found that as CKD progresses, microRNA levels decrease. That was both for microRNA 223 and for microRNA 226. No correlation were observed with inflammation or diabetes, but some correlations were found, for example, with glucose levels or hemoglobin levels. We next looked at the survival rate of patients. So that is Kaplan-Meier curves. Here you have overall survival, and below you have cardiovascular event-free survival. What we found is that when patients have lower level of microRNA 223, they have lower survival, either overall or cardiovascular event-free. And the same was true for microRNA 126. So that showed that these two microRNA, their seric levels are potential biomarkers of diseases. We compared them to the golden standard, which is eGFR, so the glomerular filtration rate, sorry. And we found that the observed link between the two microRNA seric level and mortality, cardiovascular event, and renal events was in fact dependent to the eGFR. So we have biomarkers that are as good as the golden standard, but not better for the moment. However, this does not preclude their potential role in the pathophysiology of the disease. So in conclusion, CKD is associated with a decrease in circulating microRNA levels, microRNA 223 and microRNA 126 levels. We next looked at eventual interactions between these microRNA and uremic toxins and endothelial dysfunction markers. We found that it was already shown that these toxins are increased in the blood level of the patients according to the CKD stages. So that was already demonstrated in the literature. And also, the Belgian group have shown that several endothelial markers are in fact deregulated in CKD. So MMP7, angiopoietin-1, VCAM, and CDKN1. We looked at the association of microRNA 126 with these markers. What we found is that there was a really significant correlation between CDKN1, so marker on endothelial dysfunction, on microRNA 126. We also found correlations that are already described by the Belgian group with platelet count, hemoglobin, and age. While looking at correlation between microRNA 126 and several uremic toxins, we found by using multiple linear regression analysis that microRNA 126 was associated with free endoxyl sulfate and with a total P-cresyl glucuronide. So this work that was recently published shows that microRNA 126 levels are correlated with markers of endothelial dysfunction and cardiovascular disease, further expanding knowledge on the physiopathology of this non-coding RNA in CKD. Now, how to use therapeutics. So that is in the future goals, is to modulate the expression of this microRNA in CKD patients. So that is in the future. How do you modulate a microRNA level? You have different ways. Gene therapy viruses, as we did with adenovirus in the rat. You can use nanoparticles. You can also use the CRISPR-Cas9 editing system. You can simply inject modified RNAs that are in fact chemically modified to increase their stability in the serum. And you can also use microRNA sponge binding strategy, where you sponge the microRNA and inhibit its expression. So that is my end of my talk. So I thank my collaborators, so Laurent Metzinger, and Ziad Massi, who started and are still on the project with me. The postdocs, Eleanor Bayal, Louis Clouvet, Ashraf Rangez, and so on. PhD students, Fatia Taibi, Benjamin Brigand, our nephrologist partners, Gabriel Choucroun and Philippe Fordinier, and our nephrologist partner in Belgium, Greg Glorieux, Raymond Valholder, and Eva Shepers. And I thank you for your attention.